Hello and welcome to Reviewing with Mrs. Wages. This video is intended to cover the flashback information from the DNA test. So let's begin with fermentation. Alright, what is fermentation and what is its purpose? The process of respiration always begins with glycolysis. As you remember, glycolysis needs a steady supply of NAD positive. And with the supply of NAD positive, glycolysis can make three products. These are NADH, pyruvic acid, and ATP. And this is great because the cell can then use the little bit of ATP right away. So what does the cell do with these two remaining products? Well, that depends. Is oxygen readily available? If so, the cell will follow the normal and most efficient pathway, and that is aerobic respiration. This pathway leads to the electron transport chain, which makes lots of ATP. The electron transport chain also frees up NAD positive, which can then return and be used in glycolysis. But what happens to these products if oxygen is not available? Then the cell will follow the fermentation pathway. Now, depending on the type of cell we're talking about, this could be one of two different versions of fermentation. And the result is either lactic acid or alcohol. If alcohol is produced, carbon dioxide, CO2, is also produced. But these are really just waste products for the cell. Does fermentation produce ATP? No, but fermentation of either type will free up the much needed NAD positive. This can then return to glycolysis, which will continue to produce a small amount of ATP until oxygen is more readily available for the electron transport chain. So fermentation itself doesn't produce ATP, but it frees up NAD positive so that the glycolysis process can. Okay, let's summarize all of this. The purpose of fermentation is to provide a supply of NAD positive to glycolysis in low oxygen conditions. This allows the cell to produce a bit of ATP when the electron transport chain is unavailable. So now that you understand the purpose of fermentation, let's consider the equation for each type. Let's review lactic acid fermentation first. This equation begins with pyruvate and NADH, which are, of course, the two products of glycolysis, which now serve as the reactants for fermentation. These two get converted into lactic acid and NAD positive. Now let's consider alcohol fermentation. This also begins with the same two reactants, pyruvate and NADH. Here they will be converted into ethyl alcohol, CO2, which is a gas, so think bubbles, and NAD positive. Once again, it's the NAD positive that is the most important part. Another flashback question asks you to explain crossover. So what is crossover? Crossover is the process that occurs only during prophase 1 of meiosis. It does not continue into metaphase. During this process, homologous chromosomes intertwine and form tetrads, which are the structure shown here. When crossover occurs, the two homologs literally swap sections of chromatids and thus exchange genetic information. When you look at the individual chromatids of each chromosome, you can see that they're all unique. None is exactly like the other. So what does this mean? It means that crossover, which occurs during prophase 1 of meiosis, is meant to create genetically diverse gametes. And the more genetically diverse a population is, the stronger it is. And this brings us to our next question. How many copies of a gene are found in gametes? Well, remember, gametes are haploid sex cells, which means they only have one set of chromosomes. Thus, they only carry one copy of each gene. Did you get that? Only one copy of each gene. This is necessary because gametes, such as sperm and egg cells, are meant to join together to form diploid zygotes. The The child produced by this union will then have paired chromosomes, one from each parent, and because of this, their genes will also be paired. I hope this helps, and as always, happy studying!